Hello, Dublin. Are we in the wrong place? Welcome to This Week in Virology, the weekly discussion show all about viruses. I'm Vincent Racaniello. This is episode 177, and today is the 26th of March, 2012. Today we're recording and streaming live from the spring meeting of the Society for General Microbiology in Dublin, Ireland. This is our first live streaming episode of TWiV in Europe. And we have a terrific panel today. We're going to talk about influenza H5N1 virus and new emerging bunyaviruses. And let me introduce our great panel to you, starting on my left from the University of St. Andrews, Richard Elliott.
Hi, Vincent.
Thanks for joining us, Richard.
Good to be here.
Richard and I go back a long way. We were both in the Palese lab.
A long, long time ago.
Long time ago. In the galaxy. Never mind all of that. Next to Richard from Imperial College, London, where she says she likes it, Wendy Barclay.
Hi, Vincent.
How you doing?
I'm good today, thank you.
You and I also know each other from a long time back as well.
We
right?
do.
Right.
We do.
On Wendy's left, from Erasmus Medical Center, Ron Fouchier.
Good afternoon, Vincent.
Thanks for joining us, Ron.
You're welcome.
No, you're a busy guy.
Yeah, well, we're not allowed to do any work, so...
Yeah, you have plenty of time to talk about it, right? And finally, all the way down at the end there, from Queen's University, Belfast, Connor Bamford.
Hi, Vincent.
Welcome. What do you think, your first podcast?
Yeah, it's very good, yeah. It hasn't started that much, so
All right. Well,
first 20 seconds were fun, yeah.
all of us join in the conversation as we go. So I'd like to start talking about H5N1. We have a little tight schedule here today. We'll try and touch on most of the aspects of a typical TWiV for those of you who haven't listened to one. By the way, how many of you have listened to an episode of TWiV? Raise your hand, please. Wow.
It's great.
What's that?
The weather is great.
Oh yeah, what's the temperature outside? Anybody know?
16 degrees.
15 degrees? Excellent. Almost everyone has listened to TWiV. That's just great. We usually I have different co-hosts. I have Alan Dove and Rich Condit. They're not here with me today, but when we travel, we have visitors, of course, as we do today. And we usually talk about virology, we read some email, and we do picks of the week. I have a couple of emails I've picked because they're relevant to today's discussion. I'll try and mix those in. And then at the end, Connor and I will do some science picks of the week. So let's start with influenza H5N1. Probably not a topic people talk about much lately, so we thought we would deal with this. I want to start with Ron, and we all know that you have done some experiments to study the transmission properties or the transmission correlates of influenza H5N1. And I wonder if you could just describe the goal of those experiments.
Well, the goal of our work is really to understand what it takes for flu viruses to go airborne. We know that there are many types of flu out there in animals, and most of those viruses would be zoonotic, so they can jump the species barrier into humans once and then don't spread any further. But then we know that human flu viruses, pandemic flu viruses and epidemic viruses, they spread like wildfire in humans by coughing, sneezing, talking to one another. And we want to understand what it takes for an animal virus to get this last trait by which it can then spread via talking, sneezing, and coughing between humans.
So you picked H5N1 because it's not efficiently transmitted, and then if you could get it to transmit, you could understand the correlates.
Right, we could have picked, of course, for this project just any flu virus from animals, but given that H5N1 has been a threat since 1997, given the fact that there's been so much debate whether this virus would ever get the ability to go airborne, we decided to pick this one to do fundamental science at the same time with applications potentially to prevent pandemics.
So there are other strains you could have picked which don't transmit well in ferrets that you could have also used?
Yeah, almost all avian flu viruses do not transmit in the ferret model, so we could have taken any low pathogenic duck strain, but then we could do the fundamental research, but not the direct application to human health.
So if you were to wind back the clock and do it all over again, would you pick one of these low pathogenic strains, or
No.
would you do the same thing?
I would do the exact same thing. I like to do fundamental science, and I like to serve public health with it.
Okay, so that was the goal of this work, and you use ferrets because they're a good model for influenza.
Yeah, I would say it's the best model for influenza that we have, but it's still a model. But the airways of ferrets behave pretty much the same as the airways of humans with respect to the receptors the cells in the airways express. So when cells in the human upper respiratory tract express 2,6 sialic acid, so do ferrets, and in the lower airways you have predominantly 2,3 sialic acids, which are the avian virus receptors, and we find the same in humans. And also you can replicate disease fairly well, so that when viruses are highly pathogenic in humans, they are generally also highly pathogenic in ferrets. And so it is our best model, but it's still a model.
So if you were just to take an H5N1 and make it use alpha-2,6 sialic acids, the human receptors, would that be enough to get transmission?
No, it's not. That has been published. Many people have tried it. You put in any of the mutations that were associated with the emergence of pandemic strains over the last century, those mutations do not make H5N1 transmissible. It's not sufficient. And so that's why we had to be on the lookout for what it is that makes this virus go airborne.
So with those kinds of mutations, they don't make the virus transmissible, do they allow replication in the ferret tract?
Well, the avian viruses replicate in the ferret even without those mutations. And in fact, when you change the receptor specificity, you see better attachment to the cells in the upper respiratory tract of ferrets, but you still do not see very efficient replication.
Okay.
And so back in the end of last year, we wrote an opinion article of what we thought needed to change for a bird flu virus to change into a mammal adapted virus. And that's one, it has to attach to receptors in the upper respiratory tract. Two, the polymerase proteins need to adjust so that they produce more virus. And then three, the virus needs to come out as single virus particles, not as big aggregates that cannot be transmitted by breathing. Those were just hypotheses back in the end of last year, but I think that story still holds true.
So the aggregates are determined by the neuraminidase, is that
Well, the
correct?
balance between hemagglutinin and neuraminidase, yes.
Okay. So in your experiment where you passage these viruses in ferrets, my understanding is that you first made changes to allow recognition of alpha-2,6 sialic acids. You're grinning slyly now. I guess I'm wandering into bad territory.
Well, yeah, so the National Science Advisory Board on Biosecurity has asked me to not disclose any details of the exact mutations we put in, nor of the rest of our methods, and also not disclose any of the information on the detail of the mutations that were accumulated throughout the experiment.
But no one is listening here, as you know. So you did introduce mutations initially, though. You didn't take a wild-type H5N1 and put that directly in ferrets.
We put a couple of mutations in first.
If you did take a wild-type H5N1, a very high pathogenic H5, and passaged it in ferrets, would that be an effective strategy?
No, we did that too, and others have done that also, so I can disclose that information. So you take a wild-type H5N1 and you passage it in ferrets, it doesn't do the trick.
Okay, so you made some changes to allow better replication, and then you passed it from ferret to ferret in some way. I think you probably did intranasal inoculation and took what came out of the wash and then passed it to a next ferret. I don't know the details. I don't know if you amplified it in between, but that's the general strategy. And then after 10 passages, you have a virus which is now transmissible. This is public information, right?
Well, all of it is public information. You know, we get our information from the published literature too. It's just that you don't hear it out of my mouth. So you can say whatever you want to say. I'm not going to confirm that that's the case. But after an X number of passages, but in some way in ferrets, we got an airborne H5N1.
So why do you think manual passage is a better way rather than putting animals in adjoining cages and asking for the airborne transmission to do the passage itself?
Well, what you do by repeated passage is you create an enormous quasi-species. And how fast that quasi-species really emerge, we don't know. We know now. But we decided to just go for an X number of rounds and then take this entire quasi-species and put it in a ferret and see if any particular mutants out of the quasi-species would make the jump. And the answer was yes.
Okay, so then in the end you have a virus with an altered biological property, and then you determine what sequences are responsible for that.
That's correct. We did the experiment a number of times, and then we sequenced all of those viruses back. We sequenced the initial quasi-species, and we sequenced each of the viruses that jumped. And then you will see that some mutations are in common, and some are not.
So some in common among all the different passages, right, and some are not. But the ones that are not in common don't necessarily have no relevance to transmission.
No, absolutely, absolutely. It might be that the virus can evolve in multiple different routes and
sure,
obtain the same phenotype. But we did find a number of mutations in common, and most of those were pretty interesting.
We're going to find out one day, I suppose, right?
I hope in a couple of weeks, yeah.
Oh, that would be great. So it could be that if someone did this in another lab, you would get totally different mutations.
Yes, well, so we all know that there's a second paper by Yoshiko Oka's group. He did a completely different
right,
series of
right,
experiments coming in from a completely different angle, but trying to address the same question. And I can tell you some of his mutations are the same. And that was a shock to me and to Yoshi, to see that some of our mutations are actually doing the same
sure,
thing.
So Wendy, I know you're interested in H5N1 transmission also.
Yeah.
Do you ever think of doing this kind of experiment?
Yes.
Yes? Why didn't you do it?
Okay. Several reasons. Primarily because I don't have the kinds of high containment facilities at Imperial College London that Ron and Yoshi have access to. And so it simply isn't possible or appropriate for me to attempt those experiments.
But if you did, you would do them.
Yeah, those are certainly experiments which we've thought long and hard about and considered how to do them. And we've been doing some ferret transmission work of our own in different subtypes and in different conditions. Because I think although the H5N1 work is important, as you pointed out at the beginning, there are lots of influenza viruses out there that aren't H5N1 and still haven't made the species jump. And I think there's still a lot to be learned about how they might do that as well. So I think there's a lot to do here, and as we've said as well, the mutations which make the H5N1 transmissible are varied and may go in different directions. That could be the case for lots of different flu viruses that are out there in animal reservoirs.
So Ron, the literature has a record of this, and it was originally written that the virus that you ended up with after this passage in ferrets was transmissible and virulent. And a couple of weeks ago at a ASM biodefense meeting, you reported that it was transmissible but not virulent. So I'm wondering if you could clarify.
Well, there's a lot of quotes in the press that are simply wrong.
Really?
Well, even you.
It's a joke.
I've even heard you quote me wrong in TWiV before. So this happens, you know, when the story doesn't get published, people start talking about research. The research gets better every day. What I read in the press is actually a lot more topical than what I did in our manuscript. But and of course, virulence is a tough thing to describe, because if you take this virus and you put it straight down the airways of a ferret, put it down the trachea, the ferrets drop dead. And so is the virus then virulent? Yeah, if you put it down the alveoli of a ferret, they'll drop dead, so it's virulent. But what was also in the original manuscript, and what I've also presented in Malta, is that if the ferrets receive virus by aerosol, they only get sick. They don't drop dead at all. Does that mean the virus is not virulent? No. You know, first of all, it's just a ferret. And secondly, you know, the ferrets can't really tell me how sick they are. You know, they have a ruffled fur, they have fever, they don't eat, and they are in pretty miserable condition, but they don't drop dead. And so, yeah, how sick is that? You know, if you take a pandemic H1N1, you put that straight down the lungs of a ferret at high enough dose, those ferrets drop dead also. So is pandemic H1N1 highly virulent? Well, we now do not seem to think so. But in the ferret it was, because those are immunologically naive animals. And so the real question is how would this virus do in an immune competent population that have already been pre-exposed to flu, and how severe would it then be? We can only guess, but we know that seasonal flu viruses do provide cross-protective immunity in ferrets, even against highly pathogenic H5N1.
So this illustrates 2 points. One is that if you say virulent, you have to define what you mean.
Yes.
And I teach that to my classes.
Very good.
That you have to teach the animal model, the root, the virus, everything. And press doesn't do that. They just use the word virulence. The other is that good discussions about science require publication. And I'm sure everyone agrees with me here, but it's been the problem since the beginning. You can't discuss something if you can't see it. And we're kind of hampered here, but that's the way science goes forward. So if anyone is listening, it's really important to publish everything. Ron, in a science article that you wrote not too long ago, you wrote, because H5N1 virus can acquire the ability of aerosol transmission with few mutations without significantly losing virulence, existing assumptions should no longer be used as the basis for scientific advice. So we're using virulence in a different way in that sentence?
Well, so this is really about pandemic preparedness plans, and there are many pandemic preparedness plans that go as far as a case fatality rate of 1%, some even don't go that high. There's no doubt in my mind that H5N1 does not have the supposed case fatality rate of 60%. But there are pandemic preparedness plans that don't even go up as high as 1%, which is actually even lower than Spanish flu. And so I cannot say, based on these experiments, so from these experiments one may conclude that H5N1 is a pandemic threat. You know, it's now a pandemic threat in ferrets, but it might also be in humans. And then secondly, if that virus would start to spread, what would the case fatality rate be? Right now we are unsure. But then to only set the pandemic preparedness plans for pandemics with a case fatality rate of 0.5 or 1%, I think that's a little bit tricky. Because those plans are very important when the pandemic hits. We've seen it in 2009, and that was luckily a very mild pandemic. But those plans have to be in place, otherwise all hell breaks loose.
So I'd like to get back to this transmission and virulence issue that we talked about. Were you surprised? Did you think that transmission would be equated with virulence if you passed the virus and got it able to transmit in ferrets? Did you think it would automatically be virulent? Or if you didn't, why do you think it loses virulence on passage?
My gut feeling before we started the work would be also, just like everybody else in the field, that the virulence would drop dramatically. Because the virulence now of H5N1 is determined to a large extent by the fact it causes pneumonia by binding to 2,3 receptors in the lower respiratory tract. Now, those viruses that switch receptor use to 2,6, they would be more dominating in the upper respiratory tract and have a hard time, we would think, causing a pneumonia. But if you put the virus straight down the respiratory tract, it will still give a massive pneumonia. But also in the upper respiratory tract, the ferrets are still sick. They get the flu more than they would get with a seasonal flu virus. And so they're more of a ruffled fur, higher temperature, more lethargic. And I don't know why that would be. And so I would have guessed that the virulence would have gone down, and it probably did to some extent, but not as much as I would have expected.
What do you think, Wendy?
Well, I think what we see quite often when viruses first make the species jump is that they can be quite virulent.
Right.
And that as a virus continues to evolve and adapt to its new host, the trade-off between transmission and virulence may come into play. And I think we see that with human flu, actually. When we've had subtypes of human flu that have been circulating in people for a long, long time, you see perhaps less severe flu illness. And when you get a recent jump, you see these bad pandemics. So I think we were expecting, just as Ron says, that there would be this trade-off, but I wouldn't expect to see that trade-off straight away. You know, you sort of get a gradual decrease rather than a complete loss of virulence when you go to transmissible H5.
So both of you have signed on the moratorium, the 60-day moratorium on is it all H5 research or transmission research? I don't remember.
It's research to make H5N1 transmissible. So even doing research on transmission of wild-type H5N1 still goes on.
So what is going on during this moratorium, and when it ends, what will happen?
Well, the moratorium has ended formally, right? The 20th of March was the last day. But the WHO group that assembled in February has recommended to extend the moratorium until national governments re-establish the biosafety conditions under which to do the work. And those advices, I know that several governments have looked at it, but the advices are not public yet. And so we're waiting for that essentially to recontinue the work.
So every government of the country in which there are labs doing this research is going to re-examine the containment.
That's right. So, and the Netherlands Advisory Board has said that they would stick to their old advice, BSL 3+. The ISATAC committee in the US has assembled, and I think their opinion is ready, but they're waiting for some details to be sorted on other classification issues, and they will come out with one statement very soon. And then beyond that, we need to communicate. That's what the WHO also recommended. But I think that the communication will be best done when the publications come out and we really defend what comes out, what we do with this information, what it all means. But we can only do so when the details can be discussed also.
So likely there will be some countries that can do this research at 3+ and others 4.
Yeah, Canada has classified it as 4.
Okay.
Although the definition in the Canadian version is a little bit odd. I find it hard to understand how they're going to define that. But it seems, well, I'm guessing that the US is going to stick to class 3+, and the Netherlands has said the same thing. So they still stick to the old advice and think that these experiments can be done safely under those conditions.
So you will resume these experiments?
Yeah, well, there's still a lot of debate, and so later this week we'll have another meeting with the NSABB in the US. Next week there'll be a meeting here in London with the Royal Society, at the ECMID conference, at the flu symposium. So we now really have 2 weeks of conversation, talking. And I hope by the end of those 2 weeks that both governments have made up their minds about the publication and about the research. Of course, the publication is still in the hands of the journals and the authors. So regardless of what US government and Dutch government say, the authors and the journals are going to have the last vote on the publication issue. But as far as the work is concerned, I hope in 2 weeks' time we can restart.
So
that's my hope.
WHO recommended that the work be published, but as you say, it's up to the journals and the authors before that happens?
Yes, well, it has always been like that. The NSABB brought out a non-binding advice, which was taken over by the US government. The Dutch government is investigating whether they could block my publication via export control, but it is our opinion that they cannot. And therefore, the final vote is on our side.
Would you like this work to continue?
This work absolutely needs to continue. And all of the organizations that have looked at it, including US and Dutch government, have said that this work needs to continue. The only thing they're not so sure about is the publication. But as you said, and it's my opinion too, science only moves forward by publishing, and this system of sharing information under confidentiality simply cannot be done with the current international legislation.
What do you think, Wendy?
I completely agree with that. I mean, it really scares me, the idea that there's going to be some censorship and people are going to decide based on what criteria, I'm not sure, that certain scientists can know information and others cannot. You just don't know that there's not some really bright guy sitting out there in a lab far, far away who's going to have that good idea based on the piece of information that he's not going to gain access to because somebody didn't know of him or he wasn't on a listed approved list.
I have a nice quote from your science paper, which is a good way to end this discussion. Quote, infectious disease specialists have a moral obligation to perform dual-use research in the interest of public health and to communicate the results of their work responsibly. That's very nice. I like that. It sums it up really well. Do you think it's possible to do, going forward, to do dual-use research safely and openly?
Yes, and this is really something also for this audience and also your listeners, I think, to think about. You know, if you look at the list of dual-use issues, the list of 7 by the NSABB, this is what infectious disease specialists do. They want to figure out why is one bug more virulent than the other, why is one bug resistant and the other is not. All these items that are listed in the NSABB list is the work that we do on a daily basis. If we now accept that dual-use research cannot be published, we should all end our careers, because we're not going to attract any more PhD candidates, postdocs. We're not going to get any funding. I wouldn't see how we keep infectious disease research running if we have to stick by those rules of concern and block publication of anything that might hypothetically be misused in the future. And so the only way forward, I think, is we need to do more dual-use research.
Wendy, were you surprised at the furor of the response to Ron's and Kawaoka's work?
Absolutely, because, I mean, this kind of work, in essence, has been going on for some time. If you scan back through the literature over the past 5 years or so, there are lots of papers with lots of people doing work on H5N1, trying to understand why it transmits or why it doesn't transmit. And I mean, it's only at this stage that Ron and Yoshi got successful that people have started being cross about this type of research. But this stuff is out there in the literature, and people have been thinking in this way, as we all think in this way the whole time. This is what we do.
I think either you or Ron or Yoshi should join the NSABB.
So I got invited to fly out there at the end of this meeting, actually to cut short my visit to this meeting and go and sit as an advisor on the board. And I said yes, and they gave me 2 alternate dates, but the one that they went for in the end was this one that's coming up at the end of this week, and I'd already made some considerable commitment to this meeting, so I couldn't get myself there.
I think it's important to have on that panel people working on the disease that they're investigating. And I think that wasn't the case, at least in terms of research, for the current makeup. And if they have to, they should bring in appropriate people and bring in a number that will not allow a tie to occur on any opinion. But we'll see what happens. I have an email here that actually relates to this that both Wendy and Ron could address. This is from John, who writes, in TWiV173, you talked about a study on antibody levels to bird flu H5N1 in various populations. So this was the Palese meta-analysis showing about 0.5% seropositivity in some Asian populations. And you related this to infections that don't cause serious enough illness to send someone to the hospital or perhaps to get them tested for H5N1. I was curious whether some of the people with antibody to bird flu might have been exposed to the virus but never infected. It seems like people who work with poultry could be exposed to enough virus particles that even if they don't replicate in the human body, they might still develop antibodies to them. Is this plausible? Since this is a gastrointestinal virus in birds, would droppings from infected birds be one main source of virus particles someone might be exposed to? If this explains some of the antibody against bird flu in the population, it should affect the estimate of how lethal the virus is when it does manage to infect the person. What do you think, Ron?
No, I think that's a very good possibility. But the problem is that serology is not really an exact science in flu yet. So we can't say by looking at antibodies whether somebody has been exposed to antigen only or infected. So we cannot discriminate. But it's true. So if we go back to 2003, we had a big high-path outbreak in the Netherlands by H7 virus. And there were 89 confirmed cases of infection. Most of those seroconverted. But there was the first problem. Not everybody who was infected seroconverted. So there's the first problem. The second problem is that there was very, very intense monitoring of people for clinical signs and for virus replication. And we see seroconversion in people in which we never detected virus. So what's that? But you're absolutely right. So if you go in a chicken pen, which is very dry, dusty, and there's 10 to the 8 viruses per gram of feces, and you're starting to clean up that chicken pen, then you're going to be exposed to antigen no matter what you do. And you might be developing antibodies without infection. And so I think that this whole controversy now, I think, is going to put more emphasis on doing good studies in Asia to find the real attack rate and the real case fatality rate. But right now, it is really, really hard to say how many people have been exposed, how many get sick, and how many die.
So in this outbreak you just mentioned, how many individuals got sick?
There were a couple of hundred, but this was also in flu season. So did you get sick because of H7 or seasonal flu? And so there were 89 confirmed human cases. There was one fatality. But if you then did a serological survey of all of the poultry workers, you would find 1,000 people that had antibody to H7. And that antibody assay was then set up to have a defined specificity and sensitivity based on the known infected cases. Right now, with the H5N1 studies, we often don't even have that. We don't know what the sensitivity of the assay is, the specificity of the assay. So how are you going to define a population estimate of exposure? You can't. I think that the scientists and the WHO and the national influenza centers in the outbreak countries, this is one of the tasks that should be very high on their list.
It's surprising that it hasn't been done already, or has this been an impetus to drive that forward?
This is the first time that this has really been discussed. For many years, I've been showing those numbers from the WHO website because they are the only real numbers we have. But I keep saying that this case fatality rate can't be right. It's probably way too high because all the mild cases are missed. I've been saying that for years. But I have no evidence for saying that. I'm just trying to be cautious.
Sure. Well, the press, unfortunately, only takes the WHO numbers.
Even some scientists do that.
Of course.
And the numbers are correct as long as you say your sentence in full. If you state that 60% of the WHO confirmed cases die, then you're absolutely right.
So, Wendy, what do you think of these sero surveys that have been done? Are they useless, or do they provide some information?
Well, I think they're providing us with information now. I seem to remember that there was an attempt right at the start of the H5N1 emergence back in '97 and then 2003, where groups of scientists did go out to do some sero monitoring to see how much H5 was out in Southeast Asia. And at that time, they didn't find very much. Which I think is why, for a long time, people sort of sat back and believed the WHO figures, because they kind of gone and had a look. They couldn't see that people in these areas were seropositive using the technologies that were available to them and with study numbers that were probably a little bit too small to make those conclusions from. And now on revisiting it, I think time has gone by. So we've had much more opportunity as the years have gone by for people to become exposed to those viruses and seroconvert. And I think we're probably, even now, a little bit better at detecting H5 antibodies than we were back in 1997 when H5 first came onto the radar. So we're picking more up, and there are more people who've been exposed as the years have gone by.
So, John, the rest of John's question. A related question is, if we are worried about a potential H5N1 pandemic, would it make sense to add a related strain to our yearly flu vaccine? So if it did start to spread, there would at least be some level of cross-reactive antibodies in people who'd been vaccinated. What do you think, Ron?
I don't think so. The problem there is that although we now have shown fundamentally that H5N1 can acquire the ability to go airborne between ferrets, and therefore maybe also other mammals, there are too many subtypes and variants of the subtypes out there to now put all our money on H5N1. We have been focusing on H5N1 for years, and then all of a sudden we had a H1N1 pandemic in 2009. Who would have thought that? Now, if you have a big crystal ball and you can know for sure it's H5N1, then certainly we should be producing vaccines ahead of time because our capacity is limited. But we can't. I don't think anybody has a powerful crystal ball that can guarantee that H5 is the next pandemic.
Okay, are there any Twitter questions that we need to take? 2, very good.
So the first one I've got here was, does Ron's airborne H5N1 transmit through chains of naive ferrets sufficiently? And how about if they were previously infected with seasonal flu? I'll give you the second one as well while we're here. Another one is, once everything's back to normal, what will be the next step in the research?
I'm afraid I didn't get the question 2, but I'll answer question 1 first, otherwise I'll forget what it was. So chains of transmission, we have done chains of transmission by infecting one ferret, then let it jump to the next ferret, but then we take that virus again and we let it jump again. So it's not a full free chain, there's one manual manipulation in the middle. We've not done it yet in previously exposed ferrets. That's certainly something that we want to do as soon as the moratorium ends. We have done challenges of H5N1 in previously exposed ferrets, and then you see that those animals are clinically protected, so they don't drop dead, even by intratracheal route, they hardly develop any disease. And so I'm guessing that if we would do this with our airborne H5, those animals would be perfectly healthy. But I didn't get your last question, I'm sorry.
Sorry, the second question was, once everything is back to normal after the moratorium, what will be the next stages for the research?
Right, everything back to normal, that would be nice.
For you, Ron, it will never be normal again.
Well, I hope it will. So I hope in 3 weeks we will publish, and then I go on holiday, and then after I return from holiday, it's all back to normal. Well, the things that are high on the agenda, and we were almost finished with it, is to map the minimal determinants of transmission. So we have this quasi-species, there's a lot of mutations, and we brought it back to the absolute minimum. And we were almost done when we called for the moratorium. It would be nice to see if those mutations would also confer aerosol transmissibility to other H5N1s and to other virus subtypes. So then I get to play with Wendy and put it in low pathogenic avian influenza viruses, hopefully. I would like to see by doing challenge studies to see if the vaccines, whether any of the vaccine candidates that we have around would work better against the aerosolized virus than other vaccine candidates, and the same with antiviral treatment regimens and biological response modifiers. I think these are, so far, all of our vaccines and antivirals have been evaluated with either fully humanized viruses that are not so pathogenic, or with H5N1 virus that primarily attaches in the lower airways. Now we can evaluate our options with proper candidates, and that's something that we need to do. But I really want to focus on the impact of the individual mutations and what are the biological traits that make non-transmissible viruses transmissible.
So at every time you are going to publish, will this be reviewed by the NSABB, and the possibility of redaction will always exist? No, so I think that we are now making a fundamental step in that the Dutch government and the US government are figuring out that they cannot block any publication, so it will always be the responsibility of the journals and the authors. And of course, they have to take that responsibility, and they have to communicate it carefully. Before you do this type of work, you have to think how you do it, and think how you defend this sort of work, and have a communications plan ready even when you publish to explain why you did it. And there will always be debates about particular types of research that we will have, but I think when we're done with this one for H5N1, I hope that we at least can continue quietly. I hope so. I hope you're right. We actually have another related email, which I'd like you all to weigh in on. This is from Lawrence, who he describes the New York Academy symposium that went on last month or 2 months ago. I'm interested in, he's a student in Heidelberg University, interested in influenza. I was watching the recent Academy of Sciences meeting in New York, and I wanted to thank you for the position you took at the meeting. I just could not convince myself against the idea that the real reason behind camping against the publications is anything but scientific. It might be political or whatever reason, but I feel it is not based on science. since from my small experience in virology, there are lots of experiments that might be far more dangerous than what have been done. It might be helpful for me if you could give me your thought about that. What is a more dangerous experiment? Can any of you think what might be more dangerous?
I can come up with many more dangerous experiments, but I don't want to trigger off any governments. I don't want to get any other scientists in my position.
Okay.
So I'll keep my mouth shut.
Connor, can you think of a more dangerous experiment?
Something with Ebola?
Ebola? Ebola could be more dangerous. Any bunyas more dangerous than?
Yeah, Crimean Congo hemorrhagic fever.
Yeah.
It's a nasty one. That's a tick-transmitted virus. If you deliberately went out to make that aerosol transmitted, that would be a more dangerous experiment.
Are you doing that?
No, not.
All right. What do you think, Wendy?
Well, I think people have been doing genetic engineering of viruses for a long time, and sometimes the experiments are done that change tropism in ways that people either did or didn't expect. And some of those potentially could have been more dangerous. But actually the fact is that none of those have led to any bad outcomes, well, apart from the Australian insertion of the interleukin gene.
Right.
So of all those experiments that have been done, I think some of them are more dangerous than this, but they haven't ended up with a bad outcome.
All right. Perhaps we could talk about some bunyaviruses for a bit, which is your interest, right? And there are two I wanted to touch on. The Chinese isolate from 2009, I think it was, and Schmallenberg virus. I know you're going to talk about that tomorrow, but there are many people listening that won't see that. So let's start with the severe fever with thrombocytopenia. Is that the official name?
It doesn't have an official name yet, and the name that's the working name is very unfortunate because it's very unspecific. There are a lot of viruses that cause fever and cause thrombocytopenia as well. So to tag one virus with this name, I don't think it's very helpful. But I understand there's a lot of politics going on. There's a number of rival groups in China that have come up with different names, and it'll be interesting to see what happens. This has to be formally approved by the ICTV study group, of which I'm currently chair, so it'll be an interesting discussion we're going to have about what we call this virus.
So this is at the moment limited to China, as far as we know?
Yeah, so the virus, this disease syndrome sort of emerged late 2009, 2010, and the Chinese scientists looked at known viruses that cause similar things, such as Hantan virus or Crimean Congo hemorrhagic fever virus, and it wasn't those. And then they came up with a new virus, a severe fever with thrombocytopenia syndrome virus, Huiyangshan virus, because that's the name of a mountain where one of the isolates was obtained from, or Henan fever virus is the third name that's going around at the moment.
So how is it transmitted?
This is a tick-transmitted virus. So it's occurred in people who work in rural areas of China involved on tea plantations. And so there are a lot of ticks around, and most, in fact, probably all of the cases that have resulted in hospitalization, people report tick bites. The virus RNA has been detected in two different types of hard tick, and the virus has now been isolated from one of the ticks. Mm-hmm. So although so far it's only appeared in China, the distribution of these two tick species is much wider than China. One is throughout the Far East and extends into Australia, and the other tick species is actually almost worldwide. So, this virus could pop up somewhere else.
Has it been found anywhere else yet?
No, there's one report in the literature of a case transported out of China from a migrant worker who ended up in the Middle East.
So do we have any understanding of how infection causes the disease?
Not really, not with a lot of the hemorrhagic fever viruses. It's a lot of immune response to the virus that causes, capillary leakage and vascular damage.
I suppose there isn't an animal model yet to study this.
They're looking at, the researchers in China are looking at various rodent models, so, but it's a new virus, so we're a long way to go with it.
Do we have any understanding of what animal reservoir there might be for this?
No, that's tricky. There's been some sero surveys done in the endemic areas, so a lot of livestock species showing positivity. Mm-hmm. Actually, the one species that's shown the highest seropositivity is hedgehogs. Hmm. And hedgehogs are infected by ticks, harbor ticks, so it's a possibility. But dogs is another one showing quite a high seropositivity.
You plan to work on this virus?
Yes. So the groups in China have been very reluctant to give the actual virus isolate to other labs, although I understand a number of groups in the US have requested it. So we are going to work on it, and we're doing a slightly different approach. So in our group, we developed our reverse genetics system, which means that if we have cloned DNAs of the virus genome segments, we're able to recover viruses. And so we've been supplied with the DNA clones of the virus genome, and we're now working on trying to put those into the system to recover the virus. Now, before we could do any of this work, we had to go through all the risk assessments because it's a new virus. It hadn't been classified as to what containment conditions it has to be handled in. So we've now done those risk assessments and have had approval by the UK authorities that they can be done at containment level 3. And so now we can actually start the work.
What do you plan to do? Well,
the first thing is actually try to recover the virus from the cDNAs. Mm-hmm. And one of the interests of my group is looking at ways we can actually deliberately attenuate virus replication. Because very few of those of you who were at Michel Blois' talk about Rift Valley fever. I mean, Rift is one of the few bunyaviruses for which vaccines are available. For most bunyaviruses, there's no vaccines. And so we're looking at ways of applying reverse genetics to alter the genetic structure of the virus to weaken it so that it might be used as a vaccine.
Should we be worried about this virus? Should anyone be?
Well, I think you must always be worried about these new viruses that pop up. There's a huge number of bunyaviruses around. Over 350, maybe 400 different isolates have been made. One of the things about these viruses is that they have a great propensity to undergo genetic reassortment in nature. Mm-hmm. And someone this morning asked a question about a virus called Garissa. So this is an orthobunyavirus, and it turns out that when the genetic analysis of this virus was undertaken, it was found that this is a naturally occurring reassortant virus. It has the 2 segments of its genome from Bunyam vera, which is the prototype virus, and the 3rd segment from a different bunyavirus called Batai. Neither of those viruses cause hemorrhagic fever by themselves. They only cause a mild self-limiting febrile illness, and yet this reassortant virus causes hemorrhagic fever in man. Just as an aside, when I first started working on bunyaviruses, we used to make reassortant viruses in the lab by doing co-infection experiments, and we used those techniques to map virus proteins to segments and that. And we did actually make that same reassortant virus. Luckily, no one got sick.
So you said there are about 350?
At least, yeah.
How many of those cause human disease?
Ones we know about, they range from a lot of the bunyaviruses, the orthobunyaviruses, cause possibly a mild, very self-limiting febrile illness in man. A lot of those viruses overlap in countries where malaria is endemic, so we don't actually know the true extent of infection. And I asked someone who works on malaria in Kenya about this, is it worthwhile looking to see whether these viruses are present? And he said, not really, because if someone gets a fever, they'll give them antimalarials and they'll get better. And they don't care whether they've had malaria and got better or whether they've had something else. So there's a lot of, I don't think we know the true extent of those. And then we've got the severe viruses like the hemorrhagic fever, so that's Crimean Congo hemorrhagic fever, the hantaviruses, and now this new fever virus, Rift Valley fever. And then in the US, there's a number of viruses that can cause encephalitis in kids, lacrosse, California encephalitis, etc.
So let's move on to Schmallenberg. That's also not the official name, right?
That's the proposed name, yes.
What does Schmallenberg think of that?
The residents of Schmallenberg aren't very happy because Schmallenberg's in the west of Germany, and it's one of its industries is tourism. It's a good place to go hiking and that. So they're not very happy to be associated with.
That's happened before, right?
Yeah,
That's
that reminds me of the Four Corners.
The Four Corners, yeah. So
Which is now sin nombre.
sin nombre, no name.
So what's no name in German?
Ohne Name.
What's that?
Ohne Name.
Ohne Namen. There you go. That's the name for the virus, Schmallenberg. So how was Schmallenberg discovered?
So Schmallenberg appeared late last summer, both in the Netherlands and Germany. There was a disease appeared in cattle. Cattle were basically off color. There was reduced milk yields. They had diarrhea and they showed signs of fever. Transient, lasted a couple of weeks. So all the usual suspects were tested for and ruled out, and then by metagenomic analysis, workers in Germany at the Friedrich Loeffler Institute came up with a new virus. It's an orthobunyavirus, which they then dubbed Schmallenberg. And the interesting thing is that this is a new virus for Europe. This type of virus had not been found in Europe before. There are related viruses found in Africa and Southeast Asia and Australia. So this was quite a surprise that this type of virus has appeared in Europe.
So, and what's the effect on the infected animal?
So apart from the sort of this transient illness in cattle, the virus will also infect sheep, goat, other ruminants, and bison. And generally, the adult animal doesn't show any symptoms. However, if the animal's pregnant, the virus is able to cross the placenta and then can cause severe deformities in the developing animal. So that the two major forms are either deformed limbs, which results in problems with the lambs or calves being delivered. I didn't realize this, but apparently when a lamb is born, its front legs have to go underneath its head like that, and if they're not in the right position, then it's very difficult for the ewe to deliver, and so that can result in the placenta rupturing, and you lose the ewe unless you've got someone there that can pull the animal out. And the other major deformity is brain abnormalities, where the brain doesn't develop properly. So when these animals are born, most of them die very, very quickly. If they're not stillborn, they'll die within a few minutes.
So I suppose we have no idea why replication causes these so far, right?
Not so far.
So are there any restrictions on consumption of food from farms that are infected?
No.
No?
No, and actually in the UK, Schmallenberg is not a notifiable disease. So if a farmer finds this sheep, or a bit later on in the year when the cows start calving, if the deformed animals are born, the farmer doesn't have to notify the authorities.
So you said in the UK, so did it spread there, or was it just discovered there and it was already there?
No, I think the evidence is quite good. This in nature, the virus is related to Schmallenberg. There's very nice studies on a related virus called Akabani in Japan, which predominantly spread by Culicoides midges, occasionally by mosquitoes. And the virus has now been isolated or detected in Culicoides in Europe. So most probably Culicoides midges were blown across the channel from Europe in a similar way to 2007 when we got blue tongue virus in the UK from Europe.
And where else is it now in Europe besides UK, Germany?
Luxembourg, Belgium, Italy, France, Spain. The Channel Islands had their first case announced on Friday.
So given the host range of the tick,
This is
sorry,
Culicoides midges.
Midges, sorry,
Yeah.
midges. Given the host range of midges, where could it ultimately spread?
Well, it depends on a number of things as what the local species of midge are in different countries, whether they're going to be competent and efficient vectors of this virus. We heard from Steve Higgs this morning about vector competence and how even different strains of the same mosquito species differ in their ability to transmit the same virus. The Culicoides species that have shown to be positive for Schmallenberg virus in Europe are present in the UK, so I think we can see further spread in the UK of this.
So there seems to be quite a fair amount of discussion about this. Is there a concern? It must be an agricultural concern, if anything, right?
It's an agricultural concern. It's a huge economic concern. Russia has banned all live imports of animals from Europe, and Russia is the major trading partner for the EU. It's an industry worth over 100 billion euros a year. So a blanket ban on all live animals, not just ruminants, includes pigs, is of extreme economic importance to Europe.
Sure. But in the UK, what would be the consequence of widespread infection?
The consequences are, some farmers are working on very tight limits. And in France, some sheep farms have lost 50% of their lambs from this virus. In the UK, some herds have reported 10 to 15% losses. So at that level of loss, that is a significant economic import.
So would it be feasible to do vector control to limit this infection?
Not really. I mean, there's been things like that have been tried for blue tongue virus, of trying to keep animals indoors during the midge season, but that's a long season, and there's some limited effect, but you'll never be able to.
So then a vaccine would be the most likely?
A vaccine's an option, and for the Akabani virus in Japan, they do vaccinate against this from time to time, and I think vaccines have been relatively effective there.
There's no cross protection, though, I would guess, right?
That's not been
There might.
yet.
So when would be the time frame of deciding whether to go forward with the vaccine?
I guess we now need to see what the situation in the UK is. In Europe, the lambing season is slightly ahead of the UK, so their lambing season's finished, and they're now coming into the calving season. And what we're seeing in Europe now is the decrease in new recognitions of Schmallenberg in sheep herds, but more cases coming in cows. I think in the UK, lambing is really coming. April's prime month for lambing, so we'll really get an idea of how bad it is in the UK over the next month, and then later on in the year, we'll see what's happened in the cattle population.
So are you working on this one as well?
We are working on this one. We got the virus from the German group in the beginning of February, and it's a good virus to grow.
It grows well?
It grows well in the lab, and so we just have established a reverse genetic system for that. So we'll be working hard now on trying to attenuate the virus as a possible vaccine.
Well, you got plenty of work to do, sounds like.
Keeps us occupied.
Do we have any Twitter questions on bunyas? No. No Twitter questions. All right, let me just read one more email before we move on to some picks, and this is just a nice one to read. It's from Lawrence, who writes, I just wanted to express my appreciation for your podcast and to tell you that I find your discussions fascinating. I am trained in engineering, but have a wide range of interests in all things scientific, and am now turning my attention towards microbiology. Listening to your discussions of scientific publications and hearing the scientific mindset at work is a soothing balm to my psyche in our world awash with banality, scientific illiteracy, and anti-science. I keep a notepad with me as I listen and jot down words with which I am unfamiliar and spend time afterwards researching their definitions, which opens up new horizons and broadens my perspective in areas that I likely would never have encountered were it not for your podcast. Thank you all very much for your time and effort. Just to let you see how people really enjoy learning microbiology. particularly from people who are in the field. All right, the last thing we typically do on TWiV is a pick of the week, and I asked Connor to pick something. What do you have for us?
I have a science writing competition by the Wellcome Trust and the Guardian newspaper. So if any young scientists, as long as you're not a published reporter or anything, you can enter this competition, and it will get published if you win in the newspaper, or you might win some money. So if you hear any good science during this conference, you can write about it and hopefully win yourself some money and reputation.
Are you going to join?
I might do, yeah.
You might, you should.
Most likely, yeah.
It says, join a new generation of science writers. The judges are looking for originality, bright ideas, and a distinctive writing style, 800 words. So check it out. Is there a prize? You just get to publish with
£1,000 and
no money.
The fact that you've
There is money?
published it.
£1,000. Excellent. Just like. All right, thank you for that, Connor. My pick is a blog post, and I must admit that knowing that Ron was going to be with us, I picked this. It's by David Kroll, who is a professor in the US who does blogging and is proficient with social media for advancing his field, which is pharmacology. And it's called Thoughts on Academic Scientists Giving Media Interviews. And so he says he's had a lot of contact with media, and I know you have as well. So you might want to take a look at this. Not that you've done anything wrong, but...
I've already read it.
You read it? I think it's very good. He has some interesting points, you know. He has a number of numbered paragraphs, what you should do. He's worked for quite a broad range of institutions, had a lot of contact with the press. And one of the things he says, exercise some due diligence on the reporter contacting you before being interviewed. While you can give extensive background, be prepared with a couple of nicely distilled quotes and repeat these killer quotes once or twice during the interviews. So a lot of good tips, and you should, all of you, assume that at some point you're going to talk to a journalist of some sort. And so it's really good to read this and learn how to do it. So that's by David Kroll. And that will do it for episode 177. You can find TWiV on iTunes, of course, and our website, twiv.tv, and also on microbeworld.org, where we have an app that you can use to stream the episodes to your iPhone or Android device. We also have a Facebook fan page, facebook.com/thisweekinvirology, where fans can interact with us even further. And we love getting your questions and comments. Of course, send them to twiv@twiv.tv. And I want to thank everyone for participating today, starting at the end with Connor Bamford from Queen's University. Thanks so much, Connor.
Thanks to you as well.
Did you like it?
Yeah, it was daunting at first, but yeah, it's nice up here.
All right. Connor blogs at Rule of Six. We'll put a link to that in the show notes. You should check it out. He tries to explain viruses to the world.
Tries to, yeah.
Very good. Ron Fouchier is from the Erasmus Medical Center. Thanks for joining us.
It was a pleasure.
And you're off to the NSABB.
On Thursday, leaving here on Wednesday. Yeah.
I wish you luck. I hope you prevail. Thanks for joining us. Wendy Barclay is at the Imperial College London. Thank you.
Thank you for having me.
Good to see you again.
Yes.
I last saw Wendy in a diner on Madison Avenue in New York City.
Some time ago.
It was fun, yeah. Thanks for joining us. And Richard Elliott from St. Andrews University. I saw you not too long ago.
In Brazil.
Brazil, that's right. So thanks for joining us. I want to thank the SGM for having us here. We really appreciate the opportunity to do a live TWiV here in Dublin. In particular, Paul Dupre for helping organize all of this. I don't know where Paul is, but thank you very much, Paul. I appreciate it. Also, Richard Scrace for helping out with the camera. That's him over there, right, Richard? And your co-host there on the other camera. I forgot your name, I'm sorry, but thank you very much. Ian? Thank you, Ian. And right next to him is Chris Kondayan, who is here courtesy of the American Society for Microbiology. He's head of outreach there. He is a live streaming guru. Thank you, Chris, for coming to Dublin. I know it was a hardship. Thanks so much. We couldn't live stream without him. Of course, thanks to ASM and SGM for helping get Chris here as well. I had to remind someone to do something. Paul, did you want to say a brief comment?
Not very much other than thank you.
Well, come up, you need a mic. Oh, wait, take one of those.
Not very much other than thank you very much. Social media and outreach and new ways to communicate science, new ways to love microbiology and be motivated by it. This man is a great example of that, and he's won the Peter Wildy Prize. He's going to give that talk. Stay on for that today, Vince. And all of the people who have already been thanked, and the SGM office have been tremendous in putting this together. You did us proud. Thanks a lot.
Thank you.
Thank you.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.